The reason I'm moving away from the computer and onto the blackboard is because I want to make this more interactive. I want to be able to ask you questions and have you answer. So, one thing I could ask right at the beginning, how many of you have ever run a PCR?
No.
How many of you know, okay, so we've got one person that's run a PCR. How many of you know what PCR stands for? A couple more. Okay, so for a lot of you, this is brand new. It is tied to the topic of DNA replication, because that's what the polymerase chain reaction is. It's DNA replication in vitro. So again, we're looking at the lab syllabus, page 195, application of the polymerase chain reaction in bacterial identification. So, the bacteria that we are identifying, this is going to be physiological unknown 2. And this PCR, this polymerase chain reaction, is worth points. It's worth 5 points if you do it right. If it works. So the polymerase chain reaction is a process. I was just mentioning to somebody a few moments ago, it's really important in this class to understand what's a process versus what's a thing. The only thing that's really bad, that's too vague. What's a molecule, for example? So you need to know what, when we're talking about molecules versus processes versus regulatory mechanisms, it's a good idea to try to sort it out in your head. Which one are we talking about? So the polymerase chain reaction is a process. Anybody know what it does? Some of you said yes, you've run it. What does it do? So it's a process. It's going to amplify DNA. In vitro. What does that mean? In vitro means in an artificial container, right? Not in a cell. So in vitro is not in a cell. It's going to happen in a tiny little tube. PCR tubes are really little plastic tubes. So it's a process. What do we mean by amplifying DNA? You're probably used to amplifying sound, but when you amplify sound, what happens to it? It's louder. There's more sound. So if you amplify DNA, there's more DNA. This is just making DNA. Amplifying DNA is making more. Now, the PCR is happening not in a cell, but the process that is the PCR does happen in cells all the time. So making more DNA, if this is happening in a cell, what do we call it when it's happening in the cell? Oh my word. Making more DNA.
Transcription, translation?
No. Transcription is making RNA. Translation is making protein.
DNA.
Whoa, DNA replication. DNA replication, yeah, this is asking you to actually think. And it's like, wait, what? We're trying to focus on the lab exam. And I'm saying, no, you can't do that right now. I want you to focus on this. So the process that is happening in a tube, not in a cell when you run a PCR, because it's happening in an artificial container. If you're looking at the same process happening in a cell, which is making more DNA, it's called DNA replication. That is a process, and you need to know that process. So what we've covered since we stopped talking about photosynthesis, we've talked about DNA replication, transcription, translation. Those are three very basic biological processes. Make sure you know which one is which. Do not mix them up. That's why I spent so much time talking about transcription versus translation. But don't forget this one either, replication. So DNA replication, and it requires three things, which I went over and over, because transcription requires the same three things. What's required? DNA. DNA is a template. Because DNA replication is semi-conservative. That business of two DNA strands come apart, and each DNA strand is the parent or the pattern by which you make the complementary strand. You always conserve half of it. So it's semi-conservative. That's why I said don't spend too much time thinking about how old DNA is, because it's really, really old. So you gotta have DNA as a template. What else do you have to have?
Enzymes.
Gotta have energy. And in the cell, that energy is supplied by DNTPs and RNTPs. Remember, D stands for deoxyribose, R stands for ribose. It tells you the sugar that is associated with either RNA or DNA. DNTPs are used to build DNA, but in the process of building DNA, you have to build primers. Primase has to build primers, and they're built of RNA. So DNA replication as it occurs in the cell has to have both. And what's the third thing you have to have? Enzymes. Rather than go back and talk about all the different enzymes that were required for cellular replication, let me say that for the polymerase chain reaction, there's only one. That's why it's called the polymerase chain reaction. What is DNA polymerase? It's the ending. It's an enzyme. Now, it's an enzyme used to catalyze the formation of what? Because it's very specific. You could say it's used to catalyze the formation of DNA, but I want a more specific answer than that because it's a very specific chemical reaction that takes place as catalyzed by that enzyme. What is it? Formation of phosphodiester bonds. For our purposes of the PCR, we're building DNA. We are not building RNA. The PCR, as it happens in a tube, is not the same as the cellular process. You don't have helicase, you don't have topoisomerase, you don't have primase, you don't have ligase. All you have is DNA polymerase. But it's a very specific DNA polymerase. That DNA polymerase enzyme can catalyze the formation of phosphodiester bonds where? Where? If I have a single DNA strand, so I have a series of nucleotides here. That's my template. Can DNA polymerase do anything with that? No. I'll answer that for you. No. Because DNA polymerase can only bind to a duplex. It requires a little section of double-stranded DNA, because where it can form phosphodiester bonds is at the free 3' ends of existing nucleotide chains. But it requires a duplex. I don't have a duplex. In the cell, the problem is solved by primase. Primase is an RNA polymerase. And what primase does is build a primer. Oops. Little short section of nucleic acid. My primer is here. Could have made it a different color to make this easier. However, in cells, primers are built of RNA. They're built by primase, and primase is an RNA polymerase. In the polymerase chain reaction, the primer is DNA. Notice this is primer, not primase. That's not an enzyme. A primer is a short segment of DNA. That short segment of DNA has a very specific function in the polymerase chain reaction. So let's say we have DNA, and we want to amplify that DNA. We want to run a PCR. The first thing that's going to happen is that DNA has to be induced to separate. The two strands have to come apart. Within the cell, helicase enzymes do that. They break the hydrogen bonds between the bases. But in a PCR, we can get those hydrogen bonds to break by applying heat. If you heat the DNA, it comes apart. The heat that we apply is 94 degrees centigrade. That's really hot. So it's a high enough temperature, the DNA is definitely going to come apart. You get two strands separated. We say the DNA is denatured. So when DNA is denatured, what that means really is the hydrogen bonds break and the two strands come apart. You just heat it up. The next thing that's going to happen, I have to obey all this other stuff. We're going to attach our primers. The term that's used is anneal, but what it really means is we're going to allow our primers to hybridize with the DNA. When we denature, we break hydrogen bonds. When we anneal, we're going to make hydrogen bonds. If it took heat to get the DNA to come apart, what do you suppose is going to allow anneal. We're gonna cool it. So we're gonna cool it down to 55. Actually, this temperature is dependent upon the primers themselves. But if this is our DNA that is denatured, I'm gonna move it down here. Now we have this DNA here, and we have this DNA here. And these primers are going to anneal one here, and one here. So that little short one is a primer, this little short one is a primer. And the bonds that form between the primer and the template are hydrogen bonds. Now, those primers didn't just randomly get put in those specific places, because those primers were designed to amplify a specific gene. The primers have three functions, actually. One function I mentioned up there, the primer makes the DNA a duplex. So that's a requirement. The DNA has to be a DNA duplex in order for DNA polymerase to do anything. So if we list the functions down here, they are A, make a duplex. But it's not just random. The primers are going to determine which segment of DNA is going to be amplified. A segment of DNA is a gene, so it's going to determine which gene or segment of DNA is amplified. For this particular exercise, when we run the PCR in here, as I said, well actually I said it on this board and then erased it, this is associated with the identification of physiological unknown 2. And bacteria are identified in much the same way that fungi were identified on your homework assignment. Do you remember the first question that was asked on the homework assignment? What were they using to categorize organisms? It has to do with something all cells have. And they have to have them because without them they can't make protein. All cells have ribosomes. Ribosomes are essential to cell function, therefore they can't change much over time. Consequently, the gene that we want to look at is the 16S ribosomal RNA gene. That gene has a specific nucleotide sequence. One primer that we use attaches at the front end, the other primer that we use attaches at the back end. Two opposite strands. Because the strands are not identical, they're complementary. They're also anti-parallel. This primer has a 5' end here and a 3' end here. Because DNA polymerase can only form phosphodiester bonds at the free 3' end of an existing nucleotide chain off the primer. This primer over here has a 5' end here and a 3' end here. The 3' ends have to be facing each other because we add nucleotides to the free 3' end. So the primer makes a duplex, determines which gene, which segment of DNA is amplified. It also determines the temperature, but it provides that 3' end for building on. The building being the formation of phosphodiester bonds, which is accomplished by DNA polymerase. Okay, these are the primary functions of the primer. Makes the DNA a duplex. Here's a little bitty duplex. It's going to provide that free 3' end, and it's going to determine which gene. So our primers are called bacteria 8 forward. And enteric 1530 reverse. We have one forward primer, one reverse primer. It doesn't matter which strand they're on, as long as the 3' ends are toward the middle. Once we've got our primers on there, the next step in this process is to build. We're going to add new nucleotides. So we have to raise the temperature. We're going to raise the temperature up to 72 degrees. And we're going to build new DNA. Always building in the 5' to 3' direction. That is, always adding nucleotides to the 3' end. So here we're going to build this way. And here we're going to build this way. And then we get to start over again. The polymerase chain reaction is basically that. These 3 steps. Heat, 94 degrees. Cool to 55 to anneal. Anneal the primers. Then heat up to 72 degrees. Build new DNA. Now, the enzyme involved in this process is called DNA polymerase, but it is a very specific DNA polymerase because we're making it function at 72 degrees centigrade. Think about your temperature requirements. What kind of an organism functions best at 72 degrees centigrade? Hyperthermophile. This is not just a thermophile. This is above 60, significantly above 60. These guys like it hot. So this polymerase isn't just any DNA polymerase. It's DNA polymerase from an organism called Thermus aquaticus. Thermus aquaticus is a hyperthermophile. This was found in the hot springs of Yellowstone National Park. And supposedly Kary Mullis, who's mentioned at the beginning of this exercise as the person credited with developing the polymerase chain reaction as it's typically used today, he didn't actually invent the idea of it, but he did get an enzyme that would allow this process to happen in vitro very efficiently. So he found this Thermus aquaticus bacteria, hyperthermophile, living in Yellowstone National Park. This enzyme then, this DNA polymerase, is called Taq polymerase. Taq stands for thermus, T for thermus, AQ for aquaticus. A lot of times, enzymes that come from bacteria are named in a way that's similar to that, so you know where they came from. So anyway, Taq polymerase is the primary enzyme that's used in the PCR. Actually, it's the only enzyme that's used in the PCR, and it happens to be a conjugated enzyme. What does that mean? Oh gosh. Not a simple enzyme, which means it doesn't function as a protein alone. It has to have a helper, and the helper is a cofactor. You have to have magnesium to make that thing work. So, what else can we say about this? Again, it's the same process that's happening within cells. It's just going to be happening in a tiny little tube. It happens by running through this series of events. The heat to, they're upside down now. Heat to 94 to separate, and then cool to 55 to anneal, and then heat to 72 to build, and then start the process over again. Heat, if we ended up with 2 strands up there after I finished my building process, when I heat it, both of those strands are going to come apart. If you had 1 strand of DNA, and then you double that, think about our exponential growth of bacteria, because 1 cell divides and becomes 2. DNA does the same thing. So if you had 1 strand of DNA, and you run the process, you repeat this 3 steps, In our machine, it runs about 37 times total. Take one piece of DNA, double it, and then double that, and then double that, and see what kind of a number you get if you do that 37 times. It's millions. It's millions of pieces. And they begin to get much more specific in terms of what we're amplifying, because our primers are always going to bind on this particular gene. There's nothing to stop this. It'll keep going, but then you stop it by heating it back up to 94. This one part, it doesn't go much beyond, and pretty much you're narrowing into just amplifying the 16S ribosomal RNA gene. It's pretty slick. This exercise used to be conducted by having students in a lab like this, and then there would be water baths set at different temperatures. And so people would put their reaction mixture into a tube and then dutifully carry the tubes around, and they had to put them in the 94-degree water, and then take them out and move them to the 55-degree water, put them in there, take them out, move them to the 72-degree water, put them in, take them out, go to the 94, and they had to just keep walking around. Lucky for you, that doesn't happen anymore. Instead, we have this thing over here. It kind of reminds me of a hedgehog, but this is our PCR machine. So it's going to do the temperature fluctuations all by itself. So it just keeps changing the temperatures up, down, up, down, up, down. Okay? So, what other things could I mention about this? Sometimes you'll see the word oligonucleotide in front of primer. Oligonucleotide means it's many nucleotides, but it's not big enough to be a full RNA molecule or a full DNA molecule that would be functional. It's way smaller even than the smallest microRNAs. No, it's not either. It's not way smaller, because a lot of times primers are about 19 to 20 bases, and that's how big microRNA is, about 20. So it's similar in size to that. And I don't remember specifically which one is which. We have the 8 forward and the 1530 reverse. One's 19 and one's 20. Doesn't matter that much. It does matter where they bind at either end of that 16S ribosomal RNA. And again, why are we selecting that gene? Because all bacteria have it. In fact, all organisms have ribosomes. If you were doing eukaryotic, you don't use 16S, you use 18S. Different set of primers. If you're using Archaea, they have 16S, but the nucleotide sequencers are different. You can't use the same primers. We know this because we have never, I've been doing this since 2006, and we have never amplified archaeal DNA. Never, never. So I assume it's because our primers don't do that. You have to have a different primer set. If we want to amplify fungi, we have to have a different primer set. It's 18S that we're targeting, and the primers are different. What else is on here? So one thing to think about is how this compares to the replication process. Basics are still there. You've got to have DNA as a template. You've got to have enzymes, but you only have to have one DNA polymerase, which is Taq polymerase. And you have to have energy, which is supplied by DNTPs. This is significant because, as I said, you get 5 points for doing the PCR correctly. We're going to be using a substance that's called master mix. The master mix has the Taq polymerase in it, but it also has the DNTPs. And DNTPs, as I understand it, are rather expensive to make. They're high energy molecules. You have just a certain number of them in there to build DNA. Try not to waste this stuff. The other thing I can tell you is that in a lot of laboratories, students are given little containers with different amounts of water in them, and they pretend that they're doing a PCR. This isn't pretend. You're getting a real PCR. So we let Sierra College students actually work with the molecules that you would use. You're going to extract DNA to use as a template, and then you're going to be given the Taq master mix that has the polymerase and the DNTPs, and you're going to be given the primers, which are in a little mix by themselves. And you mix the primer and the master mix and the DNA, the template DNA, and you put a little bit of water in there to make it work properly. And then it goes into the machine, and the temperatures do the rest. So basically, you get 5 points for using the pipettes correctly. That's not a small task. Remember, when you use digital pipettes, do not just push the plunger down without paying attention to where it stops. Finesse is required. And I'm going to ask you guys to work in groups for this activity so that you can kind of compare what you're doing with what other people are doing, so that if somebody has a drop of liquid that's way bigger than anybody else's, you can say, I don't think you're using that pipette properly. You can kind of monitor what everybody's doing around you. Or if you don't care if they get 5 points, just let them do whatever. Well, you can't really, because it's possible for them to suck up all of your master mix, and then you don't have it. We're really stingy with this whole operation. Well, okay. So I think that's covering pretty much the essence of how the PCR works. If you want to know what our primers look like, you can look on page 196, and the nucleotide sequences are actually shown there. Bacteria 8 forward, enteric 1530. As soon as I say that, last semester we discovered that some bacteria, the end of their 16S ribosomal RNA doesn't match our 1530 reverse primer, and we had to go back to using a primer that's called 1492. It's a universal 1492 reverse. So sometimes we do have to use that. Mostly we use 1530. So I could say 1530 reverse or 1492. If you look at those numbers, if the beginning of the gene is 0, then 8 forward is actually going to give you a start that's not quite at 0. You're missing the front end. And the other thing, if you look at these, you can see that 1530 is attaching a little bit further out than 1492. Those numbers kind of tell you where they're binding to the template. Questions? Brain dead yet?
Yes, absolutely.
Probably. This is not going to take very long. On the other hand, I don't want you to rush through it, because you want your 5 points, and that requires that you pay attention and measure things accurately.
I'm not good. Why are you bringing up your gift? Between me and Richard back there, I think we went through like
I just had a
4.
thought. I have my tubes set up for this exercise, but they're not numbered. So I have to get my numbers. Uh-oh. Actually, I have a set of numbers. They're just stuck on our physiological unknown 2. I'm a physiological unknown 1. I'm just going to transfer the numbers, and now they'll be on our physiological unknown 2. But they're not the same organisms. I left it that way.
Where's my phone charger?
I have it.
Do you? I like left it on my car and I was like, I can walk back and go get it. Or I mean just get it next week.
So,
That was a risky
kind
move.
of chill for a few moments while I get this.
I would have came back and gotten it.
I was like, I can have a little faith in her.
More than what I got.
Damn, Jay.
She loses everything. You lose
What I've
everything.
heard.
What I do is everything.
Anything about this? I believe Lucifer is on the site.
I think this is a 5.7 on here. So 26 people took 5.7.
Yeah, hard numbers are falling down.
Think we can stop this? Or you want to keep it going?
I think 2 people, because didn't we start at 10?
She said, wait, I would pause it.
Oh, is it? I apologize.
So, because the polymerase chain reaction requires DNA as a template, and as I said, this is tied to physiological unknown 2, starting your next major project, you have to get the organisms that you're going to use. So for physiological unknown 2, if you recall, what you had to do last time was select one of the number 2. You can no longer use these tubes because they're not numbered anymore, so I hope nobody needs to go back and look at physiological unknown 1. We're going to select one of the numbered tubes now from a new set, which is here. Those were slants, they were rock cultures, and they had 2 organisms each. These are not rock cultures, these are solid, and there's only 1 organism type on each one. So they are pure cultures. Anyway, so you're going to select one of the numbered tubes, make sure you record your number. So you're going to keep these tubes until I've recorded all the numbers. Eventually they'll get back in the basket, but for right now, just make sure you record your number someplace and then hang on to the tube until I get a chance to call it roll. The next thing you're going to do is you're going to transfer a 2 millimeter ball of cells using aseptic technique. Transfer a 2 millimeter ball of cells into a tube containing tris buffer. Our tris buffer is in this green box, and the tubes that you should use are the ones that don't have any markings on the top. There are some tubes in here that have numbers on the top. Please don't use them because you're going to put your number on the top. So these don't have any marking except on the side. It says tris. And it says tris 8.5. That's the pH. So there's 500 microliters in each of these little tubes. It's a little bit tricky to try to get a ball of cells off of your loop. Probably the easiest way to do it, you're going to pick up a little 2 millimeter ball of cells, put your loop in here and spin it. If you spin it, make your loop actually spin back and forth or shake back and forth, it will come off. So you want to get them into the tris buffer. This is kind of critical because you can overload, you can put too much cell material in. So if you are not certain that you know what a 2 millimeter ball looks like, we have some. So this basket has some tubes in it. And inside each one of these tubes, if you shake the tube a little bit, you can see these little glass beads. These glass beads are supposed to be a 2 millimeter bulk. Bothers me a bit because the glass beads don't all look exactly the same size. Maybe some are 2 and some are 3. The little ones are supposed to be 2. Anyway. So then, if your cells are gram positive, add glass beads. Well, how do you tell if your culture is gram positive or gram negative? You can do a gram stain. What else could you do? You could do a KOH test. That would be quicker. Or I can just mark them. How about that? Give you a bit of time. So I'll do that. Make sure I can mark them right.
Hopefully. Oh, I know.
It'd be good if I did mark the wrong one. So, if the culture is gram positive, it has a green spot. Now, the next thing that we're going to do is we're going to apply cap locks to these tubes. Cap locks are in this beaker, and they've been sterilized, but what happens as soon as you pull the aluminum foil off of this, then all the cap locks begin to be exposed to the air, and people are sticking their fingers in here and getting the cap locks off. So they're going to not be sterile very quickly, but it doesn't matter because they're not touching anything other than the outside of the tris tube. The cap locks look like little horseshoes. Look, see, it's not sterile anymore. So they look like this. Little blue horseshoe shaped things. And they have to slide onto the tris tube such that they close it. These tubes have a bevel on the cap, which makes the cap lock not fit very well. So you do the best to put it on. Can you kind of see how it's on there? Kind of, sort of. If you're in the back, it's like, really? What are we looking at there? A little horseshoe like thing that's over the front. It's trying to hold the front edge of the cap, which sticks out a little bit further. It's going to hold that to the tube so this thing can't pop open when you heat it, because that's the next thing that's going to happen. So you apply the cap lock, and then you're supposed to put your tubes in floats. So the floats come in a beaker. A float looks like this. Those holes are for receiving the tubes, but you're not going to put tubes in every single one of those holes, because the way this is set up, basically you can have a beaker of water for 3 people, or if you wanted to, you can do a beaker of water for 6 people. There's more holes than that. So either 1 beaker of water or 2 beakers of water per table. You want to fill your beaker probably about 2/3 full. You're going to have your water boiling. You're going to put your tube with the cap lock on, into this float so that the bottom part of the tube is hanging out the bottom. So they sit in here like so. What you'll find is when you start trying to put the tubes into these floats, some of these holes are smaller than others. So some of them they go into very easily, and some they don't go in easily at all. My recommendation, if you're working in smallish groups, just don't use the holes that don't fit. And then this thing sits on the water and it floats. I'll leave this one tube in here so you can see how the cap lock's sitting on that thing. So put the cap lock on, place the tubes in floats, and set these in boiling water. And you're going to boil them for 10 minutes. A 10-minute boil will open gram-negative cells because they have thin peptidoglycan walls with an outer membrane. Boiling them will cause them to lyse. They'll open up and the DNA will come out into the environment. But if your cells are gram-positive, boiling for 10 minutes will not break them. So after you do this boiling for 10 minutes, if you have gram-positive, you have to beat them. Now, some of you might be looking at this and going, okay, I'm not selecting the gram-positive because the gram-positive take longer to prep. We're not going forward with the next step until everybody's cells are prepped. So what's going to happen is we're going to start this process. Everybody's going to get tubes. Everybody's going to get cultures. And at some point, I will ask all the gram-positive cultures to be given to me, and I'm going to strap them to the top of the vortex mixers. So we use these flatbed vortex mixers. We put the tubes in on the top here, and they've got glass beads in them. We put this on maximum vibrate, and we turn this puppy on, and the beads are bouncing around in there. 2 millimeter bead, think about how big bacteria are. About 1 micron in diameter. It's kind of like putting peanuts in a jar with Volkswagens. And you bounce them around. The Volkswagens don't always hit them, but if they hit them, they're going to break. And that's all we're doing. We beat them for 10 minutes. I kid you not, there was a gal from Stanford that was talking about their procedure, and she said when they beat them, she said we beat them less than an hour. Oh my word. But it does shear the DNA a little bit, but apparently the DNA is pretty stable stuff, plus there's some repair enzymes in there, I guess. But anyway, yeah. So we will beat them soundly for 10 minutes. At the end of that, your template DNA is ready. So all of this business, physiological unknown 2, is preparation of template DNA. So we have to get this part done before we can do the PCR. So I'll roll this guy up. There are assorted beakers with floats. The tweezers for getting the floats out after they're boiling for 10 minutes. You've got your cultures here. You've got your tris tubes. Cap locks. It doesn't matter. It doesn't matter. It doesn't matter. Nobody gets a shortcut here. We all have to do this process.
Yeah, we did get off.
Yeah.
Remember, carry these by the glass and
Figure
with
it
plastic
out
cap.
before, but no, just like,
Just switch it.
Now the cat has nothing. Nothing.
Rosie?
Depends on Giannis.
So bad.
If they're gram positive, put them on ice, because what I wanna do is cool them, so that then we can put them onto the Vortex mixer to beat them. And while they're beating, I'll explain the next step, which is how you set up the PCR. So as I said, nobody's gonna get a faster start on this, because we won't get that explanation accomplished until the 10 minutes is up. So the beating part is gonna be happening. Nobody's gonna be behind on this. Unless it takes forever for you to watch.
Is there anything like on?
I know. Well, go ahead and put them in. You got bubbles coming out.
Is your guys' burner even on?
No, we're done.
Oh.
They're done.
I was looking
Do you
at.
need hot water?
No,
Yeah,
I think they're good. They just never, they have a larger volume and a smaller flame.
I was like, I don't even think they're.
Oh yeah, you put way too much water in there.
She said 2/3. It's like 2.5 thirds.
That makes sense.
That's why.
Here, Gabe, fill this.
Not hard.
You would have filled it to the top. Cause you're an overdoer.
People behind you guys.
She did say 2 minutes, it was fine.
So they're not like setting on the bottom, so they have a little bit of float.
You're gonna put them on ice and I'm gonna ask you to bring them.
No one started a timer, I'm guessing?
Oh.
Oh, you did? Okay.
You're all gonna get these. Because the numbers in here go up to 25. I have plans.
I don't know why she does that.
Yeah, so I guess if you guys are ground pasta after you put them ice.
Yeah, I was just noticing this box is sitting in there.
Go put this down, DJ.
Okay, how many people have gram positives that are finished boiling? If you have gram positives, here's what I want you to do. We're going to take the cap locks off and put the cap locks in this plastic bag up here. And bring me your gram positive cultures and I'm going to strap them onto the Vortex mixer. Not everyone is finished boiling. I can only put 7 on here at a time. There are 14 of them. Actually, there's 13 of them. Still. Bring some of the first set and I'll start strapping them. Gram positive. Leave them on ice. They stay on ice. If you're finished with your beakers, please take them off the ring stand. They are a hazard. They're like that. So as soon as they're finished boiling, take them down. So I want cool, gram-positive culture. They can have glass beads in.
Because I have to leave at 8. So I think about going about 1 and just getting 7 hours of sleep. 8 to 1. I always have some lessons on Wednesdays. It's like a class. Like I have to show up to the class. Huh?
Can't put a sign. Put a note on campus.
It worked last time before the test too, and then you came over around like 2.
Are there any more gram positives that are cool? Please bring them to me.
Nice.
Somebody has given me a tube over here. It says tube number 25. I don't know if number 25 had a green spot, but if it did, this needs glass beads in it. There are none. I need glass beads.
I was like,
I'm so
like,
You get the tube that
I know, that's what I'm saying, like I take it out and it's like,
there's home to my family, they're in the house, and I would never, ever.
Oh my god. And she's like, oh my god. Is that no the gut? She's like, oh my god.
No, the whole room, the whole room. I'm not kidding. I was just like,
Oh my god.
I can't even smell it anymore.
addict.
Oh my
I'm just so overly sensitive. Yeah, you're nose blind to a lot of things.
take off your They stay there. Can I have your attention, please? All the tubes will ultimately end up on ice. All the tubes need to have their cap locks removed. The cap locks go in this plastic bag. Once you have taken the cap lock off, put your tube on ice, unless it is a gram-positive culture, in which case bring it to me. Over here. They stay on ice. We're going to start the PCR shortly, but all the tubes stay on ice.
actually like when they thought we were gonna get out like Tuesday or something.
Oh yeah.
I gotta go over. Those are the grandpa people. I gotta go over.
What the hell?
Oh my god.
I gotta go over
I have to do like the lecture stuff for these three, and then I have to do, and then I look at, I'm like, oh, the lecture, I'm like, oh, no, lecture stuff
We're just having those conversations.
Make sure you're prioritizing like stuff too.
pictures when I get home tonight. I just do it until it fixes.
Yeah.
Then you can see exactly how
But like I'm talking about the questions, I think we laid out Oh, gotcha. Yeah, I was gonna be like, yeah.
Yeah, like the ones that are there.
You're always having your house.
Oh, different values. Pick a number. Oh, of course, but it just feels different.
Think that those little tubes are the ones that go in the just like.
So, while we're waiting for some more tubes to finish boiling, some interesting questions have come up relative to the laboratory exam in terms of what is it going to look like. You have a study guide. There's 2 examples of laboratory exam 1. Please look at them, because I pulled that up as a template and use it, modify it slightly to make my lab exam. It's going to look like the study guide. Very much so. It's on paper.
It's on paper and white.
It's on white paper.
Black writing.
Yes, with some black typing on it. Yep, yep. Very much like the ones that are there. Please. You're going to be setting up your PCR in some very little tubes. These are thin-walled plastic tubes. The PCR tubes are very small. They have a very fine plastic hinge that holds the cap to the tube. Please don't rip it off. You need the cap to be attached to your tube because that's where the number is that tells us which sample is in there. So if you guys had to use finesse before, you really have to use finesse with this one. So handle these tubes carefully, please. I'm going to make an illustration up here that is representing one of those tiny tubes magnified. So that you can see, there it is, the tiny tube. It has a little hinge and a cap. Okay? So what you're going to put into this tiny tube is a reaction mixture that includes 25 microliters of TAC master mix. The TAC master mix is TAC polymerase plus DNTP. This is expensive stuff, so please make sure that you are measuring it accurately. You're going to be using a yellow pipette for this. When you put this volume in, it should be in the bottom of the tube. So yellow pipette, yellow tip. We'll make it yellow. Now these pipettes vary in terms of what's written on them. So the yellow pipettes are over here, and the yellow pipette has a line. The line is a decimal point. The line is always a decimal point if there's a line in there. There's only a line on the yellow ones and the white ones. So 25 microliters should read 025 line 0. When you're looking at the readout, also remember that when you want to change the volume setting, because this one right now is set for 35, you must hold in the little button. It's like a clutch. Always hold in the button before you turn this thing. If you don't, it makes a very bad noise, and your instructor will scream at you. So each group is going to want one yellow pipette and one box of yellow tips. Boxes of yellow tips are easier to get from this side. So you want a box of yellow tips and a yellow pipette. The next thing you're going to add to this reaction mixture is going to be 5 microliters of primer. The good news, this is a primer mix. So it's a mix. It's 8 forward, 15, 30 reverse combined. So you don't have to measure out both of them or measure them out separately. But you're going to make a 5 microliter transfer with a much smaller pipette. These are the white pipettes. And the white pipette has a different readout. 5 microliters is going to read 0, 5, line 0, 0. Make sure you've got 2 zeros after the line, otherwise it's not 5 microliters. This one's set for 10, so these things are gonna have to be reset. The tips that you're gonna be using for the white pipettes are here. They're in a box that's purple on the bottom. When you put 5 microliters in, I want you to put it on the inside wall of the tube. It's gonna look like a spot of liquid about the size of half a BB. Anybody know what a BB looks like? Okay? Half a BB, clear liquid on the side of that. You can see it clearly, it's gonna be on the wall. The next thing you're gonna add is 5 microliters of template DNA. Now, here's a question. Do you have to change tips? Yes. Anytime you change the solution that you're using, you have to change tips. If you're working in a group of 3, you're all using the same primer mix, you can transfer the pipette from one person to the next and compare your droplets, because you want to put these on the inside wall. You want to be able to see it. This is going to be liquid in the bottom. But these, put them on the wall so you can see them. And you can kind of do a comparison to make sure that everybody's getting the right amount in there. But when you change, the template DNAs are all different, so you're going to have to change tips. It's still 5 microliters. You're still going to have this set for here, but get a new pipette for each person. That is a new tip. When you pick up the tips, remember, tap the pipette into the tip box. And don't leave your tip boxes open. That's another thing your instructor will yell at you about, because that's not aseptic. You leave them open, the tips get contaminated. And then the last thing you're adding is 15 microliters of water. Now, this last amount, again, if you set the pipette to 15, it's just going to be 015 line 0. You're still using the yellow pipette for this. As long as you don't let the pipette tip touch anything inside the tube, everybody should be able to use the same pipette. But if you accidentally touch the inside of the tube, then click off that tip and get another one. So that goes in last. So the order of entrance, this is number 1, this is number 2, this is number 3, this is number 4. Does it really matter? I don't know, but I read this. This is the sequence that you're supposed to put them in. Basically what's going to happen is you're going to have these 2 guys hanging on the wall. You might knock it in with the water. If not, what I'm going to do is gather up all of these tubes and put them into a centrifuge and do a momentary spin, which will throw all the liquid to the bottom, and then they go in the PCR machine. So, now, the tricky part of this is everything is supposed to be maintained on ice. The PCR tubes are supposed to be on ice. Your cultures, your template DNA is on ice. All the reagents are supposed to stay on ice. You have to take them out to use them, but you're not supposed to put your reagents out and leave them around at room temp. They're supposed to be cold. Right now they're really cold because they're frozen. Okay, so you guys can go ahead and get your pipettes. I'm going to come around and pass out these reagents. They're frozen. The reagent volumes that are in these tubes are rather restricted because the people that prep this lab know how much you're supposed to use, and they don't put any extra in here. But these are frozen solid, so what I recommend that you do is take these tubes and just hold them in your hand for a little bit to make sure they're liquid, so you can actually use them. Or you can roll them between the palms of your hand.
Got that?
We're all
Of these? We should. You
just do a different tip for each one.
Did somebody time that 10-minute vortex?
I did.
Is it done?
This one off.
Wow.
Literally just. Oh, the fireworks.
I like that they did that.
The water's in there too.
You know what I've been doing all day?
Sitting.
Actually, no, I volunteered at the hospital today.
No.
Yesterday, I sat all day and studied.
Right. So they're all frozen.
I really need to be able to check my email right now.
Okay.
Are you recording numbers? Oh, I'm sorry, we have one. Oh, you got all.
Yeah, I think everybody got them all.
We didn't get ours.
Apparently we only got them in the front.
Okay, these large tubes that are up here are template DNA from the first vortex mixer. You will probably need to reapply your numbers. Most of them are readable, so you're good.
What? Those are the first ones.
Oh, that's the first one.
Yeah, the first batch.
Are you for certain?
Yeah, she just said this is from the first batch.
You're 5.